Yeah. So I think the last talk already did the introduction and covered all the background needed for this talk. So I'll get started. As you saw in the last talk already, that the latest long and accurate sequencing technologies, especially from 2020 onwards, with the advent of Bioinformatics, nanopore, duplex, and so on, they have allowed genome assemblers to reconstruct both paternal and maternal haplotypes. Right? So basically, for genome assembly, especially for human genome assembly, the collapsed 3 billion base pair sequence is no longer the state of the art. Now, the expectation from a genome assembler is that it should produce a haplotype resolved genome assembly containing 6 billion characters. Now, if I go back to the fundamental data structures that we use for genome assemblies, the popular data structures are obviously the Bruijn graphs, overlap graphs, and string graphs. Right? So, as we have already seen in several presentations, the Bruijn graphs are nothing but where vertices correspond to the k-mers and edges imply suffix prefix of overlap of length k-1. In overlap graphs, here you have vertices corresponding to each read segment. So each read corresponds to a vertex in the overlap graph, and the suffix prefix overlaps between reads correspond to the edges in the overlap graph. Finally, the third data structure that I want to mention is the string graph, which is was developed by Myers. And the definition of string graph is that it's a subgraph of the overlap graph. How is it a subgraph? So when we talk about string graph, basically, which is the now used in the state-of-the-art assemblers, string graph is a subgraph of the overlap graph in the following way. So it only keeps the longest suffix prefix overlap between a pair of reads, because if a pair of reads have multiple suffix prefix overlaps, it will only keep the longest suffix prefix overlap between the pair of reads. Second is, it will remove all the contained reads. So if there's a short read which is contained in a longer read, then the shorter read will be thrown away and it will be marked as redundant in the data. Third step is that it will remove the transitive edges as well. So for example, if you have 3 reads where you have overlap from read 1 to read 2 and you have overlap from read 2 to read 3 as well. So then the edge from read 1 to read 3 will be deleted, because the same path that can be spelled from read 1 to read 3 can also be spelled by going from read 1 to read 2 and from read 2 to read 3. So in summary, string graph is a subgraph of overlap graph, and it involves these sparsification techniques to make the graph simpler. Now for those who don't work in genome assembly, here is a quick example of a string graph constructed from similarly reads from E. coli genome. So here the entity read length is about 40,000 base pair. Right? So the vertices correspond to the reads themselves, and the overlaps indicate suffix prefix over matches of length at least 1,000. So as you can see, once we remove all the contained reads and we remove the transitive edges, what you get is a nice clean graph that spells the entire genome with only 159 vertices and 158 edges. If we don't apply these cleaning steps, what you have is a messy graph, right, with significantly many number of vertices and significantly many more edges, right? So I would argue that without these steps, basically this graph is in some sense useless to an end user, because the length of heuristics is very small in such a graph. Now, why am I discussing these sparsification techniques? So when it comes to genome assembly, one of the fundamental questions that one can ask is, suppose I give you input reads that cover the entire genome. Do we have a guarantee that the true chromosome can be spelled as a block in the graph? Right? So that's a fundamental guarantee that you would want from a graph model. First, I need to convert this question into a well-formulated problem to be able to answer it. Because this true chromosome is not a well-defined term, because this is not known a priori. So to convert this into a well-formulated question, what we do is, we instead ask, suppose the input reads cover the entire genome, do we have a guarantee that each candidate chromosome can be spelled as a block in the graph? Now what do I mean by candidate chromosomes? Basically, here's a quick example to indicate what a candidate chromosome means. So suppose I give you two reads given here, then you have two candidate chromosomes possible from these reads. You can spell this chromosome, or you can also spell these chromosomes from these reads. So because it is not known a priori which of these chromosomes is going to be the true assembly, we ask this question in terms of saying that do we have a guarantee that each candidate chromosome can be spelled as a box in the graph. More formally, so I have to define this candidate chromosome. So formally we can define a circular string Z as a candidate if there exist two parameters L1, L2 such that all intervals of length L1 in string Z include the starting position of at least one read of length L2, where L2 is greater than L1. So intuitively, what we just want is that the reads should cover that candidate and consecutive reads should have some overlap. That's what this definition is saying, right? But now this question is now a well-defined question that we can ask. So with this definition, we did a rigorous analysis of the three graph models, and we proved that the Bruijn graph model is guaranteed to be coverage preserving if the parameter k, which is the k-word length, is less than equal to L2 minus L1 plus 1. For overlap graph, the guarantee is preserved, that it is guaranteed to be coverage preserving as long as the minimum overlap length that I use to construct this graph is less than equal to L2 minus L1. However, for string graphs, which is the most commonly used data structure in genome assemblers, this guarantee does not hold for any value of k, where k indicates the minimum suffix-prefix overlap length. So, for all these three, we have proof, right? So, for the Bruijn graph, we prove this by contradiction. For overlap graph, I'll show just a proof sketch, and but for string graph, I just need to show you a counter example. So, let me show you just the proof sketch of how we prove these statements. So for Bruijn graph, I just need a simple proof by contradiction, where I can assume that there is a candidate string Z which is not spelled by the Bruijn graph. Now that basically means that there has to be at least one k-mer in my candidate string Z which is absent from the set of vertices in the Bruijn graph. Now with the assumptions made that the candidate string must be covered by the reads, so there must be an interval of length L1 which has a read of length L2 starting in that interval. Now if I identify the first k-mer in the candidate which is missing in the graph, I can prove by contradiction that that cannot happen because this interval must have, this read must have that k-mer in it. So for Bruijn graph, it is guaranteed to be coverage preserving. Next, for overlap graph, we have to be slightly more careful because now the vertex label lengths in overlap graphs are variable, and even the suffix prefix, the overlap lengths are now variable, unlike a Bruijn graph. So we need to be more careful, and here the proof technique that we used is that we gave an algorithm that if you give me a candidate string, I'll give you a walk in the graph that spells the candidate string. So if you give me a candidate string Z, basically what I'll do is the algorithm will identify all the intervals in that candidate. It will pull out the reads that cover that candidate string Z, and from these reads we will identify a subset of those reads which have a proper suffix-prefix overlap among them. So once we do that, we know that there is a path or there is a walk in the graph that will spell the circular string Z. Right? So that proves for the overlap graph. Finally, as I mentioned, for string graph, the guarantee does not hold, and this is easily clear from this counter example. So if we have 4 reads, let's say, from these 4 reads, I can construct 2 candidate chromosomes, right? But note that the red read is contained in the green read. So if I remove the red read, then this first candidate cannot be spelled in the graph anymore, because this blue read will now be disconnected, and this coverage gap now occurs in the graph. So this is just a theoretical assessment. So what are the follow-up questions that we can answer? First is that, does this theory affect practice? Does this limitation of string graph affect the genome assembly quality in practice today? Second, because we have seen that string graph loses this coverage, is there any alternative model that one can use that is also practical as well as provably good? Third, this problem in string graph is occurring because of the contained reads. Are there any good heuristics which we can employ to recover the non-redundant contained reads that are necessary to avoid the coverage gaps? So first, we look at safe rules that we can define to sparsify these overlap graphs. The first is obvious, that one can remove a vertex or an edge in the graph if the set of circular string walks remain unchanged. Right? So that's an obvious step, because then if all the set of circular string walks remain unchanged, then all the chromosomes can still be spread in the graph. The second is that the transitive edge reduction from Myers is also guaranteed to be safe. Third is that removing a contained read is safe if it maps to a single candidate genome at a unique location. So if there is a contained read which I know if it maps to only a single candidate sequence, then I can still remove that contained read from my analysis. So these are the 3 provably good techniques we have. Unfortunately, only the transitive edge reduction from Myers is practical among the 3, because we don't have any efficient polynomial time algorithm to implement first and third steps. For example, enumerating all possible string walks in large assembly graphs at chromosome scale is a computationally challenging problem. So we don't have practical methods, but these are the safe rules that we have. So then we have to ask, what are the heuristics that can be implemented to get this done? So these heuristics are motivated by the safe rules. First is that, as I mentioned, if a contained read maps to a single candidate, that can actually be detected in practice if you look at the read all to a read overlaps among reads. And if we see a heterozygous variant in the contained read, so if the contained read has a heterozygous variant, then I know that the contained read must correspond to a single haplotype. And if the coverage of the all read overlaps is not significantly high, then I also know that the contained read should map to a single location in my true genome. So this heuristic is employed to detect the contained reads which can be removed safely from the analysis. Second heuristic is that because we cannot enumerate all the chromosome length walks in the graph, we can at least look at the local neighborhood of those contained reads and check if the presence of k-mers actually informs that there is a new walk that can be spelled by the contained read. So here we go to the graph, we look at the contained read and extract the k-mers from the graph in the vicinity of that contained read. If all the k-mers are also observed from the parent reads as well, then we know that it is unlikely that the contained read contributes a new walk in the graph. So again, if this condition holds, we remove the contained read. Otherwise, the contained reads are retained. So the actual implementation looks like the following. We call it contain X, which takes the long reads and all-to-all read alignments. It first of all applies heuristic 1. Basically, we discard contained reads which have X snips. We build overlap graph, apply transitive edge reduction. We discard contained reads which lack novel walks using k-mer-based heuristic. And finally, we output the contained reads which we think are important for genome assembly. So let's see how this method works in practice. So for testing this, we simulated error-free long reads, but the reads have actual real read length distribution. So for example, if I simulate nanopore reads, then the nanopore reads will have skewed read length distribution, whereas hi-fi reads will have the normal distribution like hi-fi. We are using two target genomes. One is the haploid CHM13 genome, and second is the HG002 diploid genome. And the reason for using similar data is that now, because we know the ground truth, the target genomes, we can also evaluate where the coverage gaps occur by removing contained reads. So first I want to show you how many coverage gaps happen by removing contained reads that all genome assemblers do. So first scenario is the haploid genome, the CHM13 genome, where the second column shows you that the count of contained reads in this data set is in millions. It turns out that for haploid genome, there are no coverage gaps caused by removing contained reads empirically. But I want to remind you that in theory this can happen even in haploid genome, where if you have an identical repeat in the genome, then suppose there is a read 3 which is contained in read 6 because of the identical repeat. Removing this read 3 will cause a coverage gap in the second repeat. So in theory, coverage gap can be reduced, but in practice we are observing that zero coverage gaps are being introduced, maybe because there are not that many identical repeats, long identical repeats in the haploid genome. However, the story is different for diploid genomes. For diploid genomes, we see coverage gaps for both nanopore and hi-fi datasets. For nanopore, the count of coverage gaps is significantly higher relative to hi-fi. So here we see on an average 50 coverage gaps for the two datasets, whereas for hi-fi we observe only one dataset, one coverage gap across the entire human genome. Now at this point I want to remind you that for diploid genome, the coverage gaps can also happen because if a read, because what can happen is if there is a long read sequence from one haplotype, it can knock out reads from other haplotype if the reads in other haplotype are all short. So here, if you have haplotype 1, the read number 3 will knock out read number 9, and it will cause a coverage gap in the diploid genome. Right? So this is the first sort of evidence, empirical evidence, and a quantified way of predicting how many coverage gaps are introduced in diploid data. And the reason for observing more coverage gaps in nanopore data is obvious, because this nanopore follows a skewed read length distribution. So the chances that a read is contained in a longer read is much higher for nanopore data compared to hi-fi. Finally, we evaluate the method that I just proposed, the heuristics that recover the non-redundant contained reads. And we compare against three other methods. So first is extreme policy, where you retain all the contained reads in the data during assembly. The other extreme policy is that you remove all contained reads, as the string graph does. And then there is a method proposed in a 2016 paper, which has another technique to recover contained reads. So what we see here is, so first of all, the first column indicates the count of contained reads retained. The second column indicates the count of junction reads in the graph. That indicates the number of reads that have either more than one incoming degree or more than one outgoing degree, just to indicate how tangled the graph is. So of course, if you retain all contained reads, the graph will be most tangled, right? So here, if you see, the conclusion here is that the containance method gives a good trade-off between the number of junction reads and the number of unresolved gaps that are there in the genome. So here, the number of junction reads is comparatively significant if you compare to retaining all contained reads, but the count of gaps is significantly fewer if you were to remove all contained reads. We also compare against hi-fi ASM. Hi-fi ASM has a heuristic where it will recover the contained reads if it connects two contigs in the end. So it does it at the post-processing step. And here we see that HiFi-SM, because it's an end-to-end assembler, it has more sparsification heuristics, but still it has coverage gaps left in the final genome assembly. So that indicates that there is promise of improving the existing algorithms and using a better graph sparsification model. So finally, I'll conclude by saying that the provably group would assembly graph models will be useful for accurate diploid human genome reconstruction. I showed you that the commonly used string graph data structure is not coverage preserving both in theory and practice. Especially, my results were only focused on diploid genomes, but if you go to polyploid genomes or cancer genomes, this issue is more likely to occur because there you have multiple copies, and in case of cancer, you have mixed populations. Finally, I also say that the optimal sparsification of overlap graphs remains unsolved. In this work, we proposed safe rules and two promising heuristics to address this problem. So, yeah, with that, I'll thank you all and happy to take questions.
Are there questions?
Thank you, Chirag. Great talk. Is there any, based on what happens when you remove the contained reads, is there any value in the overlapping algorithm to giving the overlapping algorithm some sort of hint as to what, say you have the parent read, and you give the overlapping algorithm a hint as to what contained reads were there, then get rid of the contained reads and let the overlapper somehow just do enough work to not miss the overlaps that it would have found given the contained read. If that makes any sense. In other words, there's a parent read, every contained read is just some substring. If you gave the overlapper the whole package, here's the parent read and here's the collection of contained substrings, you clearly won't have lost any information, but it might still be a more compact way of expressing the problem.
Right. So you're right that if you preserve the relationship between contained reads and parent reads, we don't lose any information. The question is, how do we integrate that in the graph? Like, do we want to have vertices correspond to the contained reads, or do you want to remove the vertices corresponding to the contained read? So yeah, if we include them, the graph becomes complex. If we exclude them, then we don't have the guarantee anymore.
Right.
Yeah.
Yeah, thank you.
Questions? Hi, other questions? I guess in turn part, you on one hand assume that the reads have no errors in them, but on the other hand, you assume that there could be reads which do not fit anywhere in the chromosome. So, can you somehow handle the errors, and on the other hand, somehow maybe require it to more respect and the normal reads have to be used?
Yes. So the theoretical analysis is done with error-free reads, and but the second property you mentioned, that we have, like in the formulation, we make sure that each read is sampled from at least one candidate chromosome. So there's no read that comes out of magically.
Okay.
Right. So that assumption is being made.
Maybe I have one more. So, as far as I understand, the assemblers also need to have some sufficient coverage, and this classification can also, maybe if there is one lead, it can also not be used in the final result. So, can you please tell me?
Yes. So yeah, you are right that coverage gap, another source of coverage gap is low coverage data. But I mean, so I would say from a data structure side, right, that's a problem of missing data, right? So that is okay for an algorithm to not produce a good assembly. But I guess my viewpoint here is that if there is data and there is uniform coverage, then at least the algorithm should guarantee that it does not miss that region. But in practice, you are right that because of non-uniform coverage, there may be additional gaps, especially when you are sequencing at low coverage.
Yeah, but I'm more thinking about like if you have one more read and then many contain ones that have sufficient coverage, but if you remove them, even
especially during the read overlapping stage, they do include contained reads in that step. Because if there's a read and it's not overlapping to anything else, then the assembler would remove the read, marking it as primary. But if, like the figure I showed here, basically, so yeah, this one. So in this part, the contained reads will be included during error correction stages as well as estimating chimeric reads and so on. It's only when the assembly stage starts, then the contained reads are drawn.